Assalamualaikum everyone, I am Shehvar Zara. I hope you all are fine. Today our topic of discussion is modification and processing of messenger RNA. Before we start our today's topic, let's take a brief overview of the things that we have discussed in our previous lecture. So in our last lecture, the lecture number 8, we discussed in detail the chemical modifications that takes place at the two ends of eukaryotic messenger RNA, that were the addition of 7-methyl guanine cap at the 5' end and the addition of poly-A tail at the 3' end of the messenger RNA. In today's lecture, we will discuss purification of messenger RNA, and later on we will discuss in detail the third step of post-transcriptional modification, that is intron splicing. So let's start our topic. So in last lecture I told you that how the poly-A tail is being added step by step to the 3' end. Now let's see that how this poly-A tail, that is the part of most of eukaryotic messenger RNA, it provides a convenient method or provides a convenient mean for the purification of these RNA. So the technique that is being used for this purification process is affinity chromatography. Affinity chromatography is actually a special type of chromatography in which a solid matrix is being used, and this solid matrix, it carries reactive groups to which the molecules that are to be purified, they bind selectively. Means the molecules that you have to isolate, they interact with this solid matrix and they get isolated, means they get separated. In case of purification of messenger RNA, the solid matrix, it carries basically poly-DT or poly-U. Poly-DT is basically the abbreviation of thymidine homopolymer phosphorothioate, whereas U is the uracil. So now let's see that how this purification process takes place step by step. Here you can see this solid matrix where it is written poly-U. I have told you that in case of messenger RNA, the poly-U is being used at the solid matrix. So what we do is that we apply the total cell RNA. Total cell RNA means that this RNA contains all three forms of RNA, that is the messenger RNA, transfer RNA and the ribosomal RNA. Then what happens is that because the messenger RNA, it has poly-A tail, so it binds with the solid matrix. You can see here it is being shown. But this ribosomal RNA and this transfer RNA, they do not have poly-A tail, so they do not bind with this solid matrix. Here this binding is being shown between U and A. Next what happens is that once this messenger RNA binds, you can separate this by washing with the high salt concentration solution. This high salt concentration solution is basically 1.5 molar NaCl, and this NaCl, what it do is that it destabilizes the base pairing between this solid matrix that was of uracil and this A. You can see here this when we use this A means this messenger RNA, it gets separated and you can get this messenger RNA in the purified form. So this was all about the process of purification of messenger RNA. Now we will discuss in detail the third step of transcriptional modification, that is intron splicing. So introns were first studied in 1970s, and introns are basically the non-coding regions of the gene, whereas exon, they are the coding regions. So intron splicing means that before the translation takes place, these introns, they have to be removed from the sequence, and the exons, they have to be spliced together, means they have to be joined together. There are 4 types of introns. You can see in the slide. The first type is the introns that are present in the protein coding genes, and these introns, they are removed by spliceosomes. Spliceosomes are basically complexes that are composed of small nuclear RNA, and these spliceosomes, they remove introns in protein coding genes. We will discuss the structure of spliceosome in the coming slides. The next type is the introns that are present in transfer RNA genes, and these introns, they are being removed by the proteins. The third is self-splicing introns, and these are basically the group 1 introns. And what these introns do is that they catalyze their own removal from the messenger RNA, transfer RNA and the ribosomal RNA using the guanosine 5 triphosphate. You can see there it is written GTP. So these introns, they use GTP to self-splice themselves. Then you have last type, that is group 2 introns. These group 2 introns, they are also self-splicing introns, but they do not require GTP in order to remove themselves. So these were the 4 types of introns. Here is the structure of the spliceosome. Each spliceosome is basically composed of 5 subunits called SNR and PS. It is written in the slide, you can see it is marked. So these SNR and PS is basically abbreviation of small nuclear ribonuclear particles, and it is being pronounced as NURBS. So what these spliceosomes do is that they result in the splicing together of the 2 exons, and they release the introns in the lariat form. You can see here in the slide that this is intron, okay, this is exon, and this is that SNR and PS. So what these spliceosomes do, it removes the intron in the lariat form, this is the lariat form, and it supplies together the exon. You can see these 2 exons have been joined. So this was the structure of the spliceosome and how it joins the exon together. So now let's see that how this splicing of introns takes place. While we are discussing this intron splicing, so an error always occurs while discussing this process, because there are 4 types of introns, as I have told you before, and all these 4 types of intron, each of them have their own specific, or you can say special features, and as a result each has different splicing mechanism. So this causes an error while studying these. In today's lecture we will discuss the intron splicing of the GTAG introns. And the reason to discuss this GTAG class of introns is that because all the introns that are found in nuclear gene that are transcribed into messenger RNA, they fall in this category. So that's why we are discussing this. So this GTAG name, it basically refers to the fact that virtually all the members of this class of introns, they possess the same dinucleotide sequences at the 5' and the 3' end. So this name of GTAG, it basically signifies this, okay. GT here is basically the first 2 nucleotides of the intron, and GT is of course the sequence in the DNA, because you know that in DNA thymine is present, whereas in RNA it is basically the uracil. So in the transcript it will be GU instead of GT. And AG is the last 2 nucleotides of the intron. And this GTAG is commonly known as GTAG rule, and if you will like search anywhere, it will be like you will find the result of GTAG rule. So it is also known as this, and it's basically ये जो GTAG है आपके पास, ये represent करता है smaller portion को of a longer sequence, because the actual consensus sequence you have is the longer one, and this GTAG represents the smaller one, and this consensus sequence, it is basically found at the 5' and the 3' splice junction. Splice junction means the point where the splicing will take place. So this GTAG is the small portion of that region. And if you see in vertebrates, then the full consensus sequence, which I am telling you that this GT and AG is basically the representative of the longer sequence. So in vertebrates, this full consensus sequence is, for 5' site it is 5' AGG TAAGT 3', and this 3' slice site in vertebrates is 5' PY 6 times PY and CAG 3'. Okay, so here this PY in the 3' splice site is basically indicates the position where either of the 2 pyridine molecules can occur. Okay, so this PY is basically the abbreviation of pyridine, and 2 molecules of pyridine, either of the 2, means either one will, like either C or either U will attach here, will occur here, and N is represents any nucleotide. And this consensus sequence you can see here, if you study it, of 3' splice site I am talking about. So it is basically 6 pyridines followed by the nucleotide, and then followed by the C, and then the AG. Okay, and this AG, you can see I have marked it with arrows. So these arrows represent the site where the splicing will take place. Okay, these are which I have told you that the splicing junction. So this the arrows you can see between A and G and between that 3 prime, so it is basically the splicing sites. One thing you have to keep this in mind is that these sequencing, they are basically consensus sequencing. Consensus sequencings are the sequence of DNA having similar structure and function in different organisms. But here you have to keep this in mind that these sequences might differ slightly in the individual introns, because these are the consensus sequences and the basic structure may slightly differ. But this GT and AG introns or the components, they will be similar, they will almost always present. Okay, now so the biologists, I have told you that the entire sequence will be same in the last slide, except for a few changes, but the GC and AG will be present. So it was considered that the sequencing that is present around the 2 splice sites, they were originally thought to be the only regions of nucleotide similarity, GT AG introns. ओके, सिर्फ यही समझा जाता था कि जो आपके स्प्लाइसिंग साइट्स के ऊपर जो सीक्वेंसेस हैं, दे हैव न्यूक्लियोटाइड सिमिलरिटी ऑफ GT AG. ओके, बट यीस्ट में क्या हुआ कि इन G Saccharomyces cerevisiae देयर इज़ एन एडिशनल कंजर्व्ड सीक्वेंस, एंड दैट एडिशनल कंजर्व्ड सीक्वेंस इज़ बेसिकली द TAC box, and that sequence is 5 prime to, you can see it is written in red, TAC 3 prime. Okay, so this is additional sequence that is being found in the yeast, and this sequence is exactly the same in all yeast introns that have been studied so far, and it is positioned within the introns between the 18 and 140 base pair upstream of the 3 splice site. Okay, ठीक है, तो ये जो डिफरेंस था ना, ये दिस डिफरेंस, इट वाज द ओनली डिफरेंस दैट वाज बीइंग फाउंड इन द यीस्ट. Next you have uRNAs. uRNAs are involved in splicing of GT-AG introns, and these are special type of RNA molecules that are being involved in intron splicing, and this U means that these are uracil rich RNAs, and their other name of this RNAs is small nuclear RNAs, that is being abbreviated as snRNAs. So where it is written that snRNAs, it means small nuclear RNAs. There are different number of uRNAs in vertebrate nuclei, and the most abundant of them are being U1 to U6, and these uRNA are quite short, and they vary in size from 106 nucleotides for U6 and 217 nucleotides for U3. So these their length varies, and the sequence of each type of uRNA is similar in all higher eukaryotes, and analogous molecules are present in lower eukaryotes such as yeast. uRNAs are stable RNAs, and they are as important as the ribosomal and the transfer RNAs that are found in eukaryotes. And in the cell, uRNAs exists in special particles, or you can say in particles called the small nuclear ribonucleoproteins, snRNPs. Okay, so they are in eukaryotes, they are found in small nuclear ribonucleoproteins. So this was all about this uRNAs. So let's discuss the splicing pathway for GT-AG introns. In recent years, the experimental studies, they have enabled us to understand this pathway by developing procedures for purifying unspliced transcripts and subjecting them to splicing under controlled conditions. So as a result, we are enabled to understand the entire pathway. So here in the diagram you can see the process. We will discuss the steps one by one. So the first step in the splicing pathway for GT-AG intron is that the cleavage occurs at the 5 splice sites. You can see here that the 5 splice site is being marked. Here you can see this one. Okay, this is 5 splice site, and here the cleavage takes place. So the first step is the cleavage of 5 splice site. Okay, now let's discuss the next steps. So before we discuss these steps, first I want to tell you here that in introns there are two different sites. One is donor site, that is your 5' end of the intron, and other is your acceptor site, that is the 3' end of the intron, and it is required for splicing. Whereas there is an other portion or other you can say the site as well, that is the branch site, and this branch site is it is near the 3' end of the intron. So these are the three sites that are present in the intron. So we will be discussing, I will be seeing these terms, so I hope that it is clear to you that what these sites are. So the second step after the cleavage of the 5' splice site is that you can see here this you can see that after this cleavage, this 5' end, it is this resulting 5', it is free, and what happened to this free 5' end, it happens that this end is attached to an internal site within the intron to form a lariat structure, and here you can see this is the lariat structure. This one folds around and it forms a lariat structure. This structure is known as lariat structure, and lariat structure is basically formed when the 5' G of the intron, it is joined in a 2' 5' phosphodiester bond to an adenosine near the 3' end of the intron. So you can see here that this phosphodiester bond, it is being formed between the G, here it was G, so this G, it attaches with the A, and this A, which I have told you, it is near the 3' end. So this is basically the lariat structure. However, in the yeast, the branch site which I have told you is basically the site near the end of the 3' end of the intron. Branch site is the last A in the TAAC box. I have told you that in yeast there is an additional conserved sequence, that is this box TAAC box. So in yeast the branch site is the last A in this box, and in vertebrates the branch site is also an A, but as vertebrates in introns they do not have this TAAC box that is present in yeast. It is not clear how this A is selected, so it is not clear yet that how this A, specific A, is being selected in this process. The third step of this intron splicing is that the 3' splice site, it is cleaved and the two exons are joined. Okay, so here you can see this cleavage of 3' splice site takes place, and the exon ligation takes place. So here the two exons, they join together, means 5' this end, I will tell you here, this end and this end, they both will join together, and this is basically the entire process of intron splicing of GTAG introns. So now the question arises that how the small nuclear RNAs, they are being involved in this process of splicing, and what happens next. So we will discuss these steps in the next slide. So now the first question, that how the small nuclear RNAs are involved in this process. A lot of research has been done to understand this process. So it has been known for the long time that the U1 RNA, you can see here it is being marked here, like this is U1 RNA. So this U1 RNA, it is able to base pair with the consensus sequence around the 5' splice site. You can see here this is the 5' splice site. I have told you that in eukaryotes nuclei, U1 to U RNAs are present, and they are from U1 to U6. Sorry. So U1 RNA, it is able to base pair with the consensus sequence along the 5' splice site. You can see in the diagram, and this U1 SNRNP's attachment to this region of the intron, it is thought to be the one of the first step in the splicing of the intron. So this basically attachment of U1 to the 5' splice site is the first step in the splicing process. So what happens next is that once the U1 RNP is being attached to the 5' splice site, then at the branch site, which is near the 3' end, the U2 snRP, it attaches itself to that branch site, and here too the base pairing is being involved, but it is considered that this base pairing actually takes place between the U2 snRNPs, that is single small nuclear RNPs, rather than the protein that is present within this snRNPs, rather than the U2 RNA itself. So it is considered that base pairing basically takes place between the protein of U2 and the branch site. What happens next is that it has been suggested that the U1 and the U2 snRNPs, they have affinity for each other, and as a result of these affinities, they draw the 5' splice site towards the branch point. And the other snRNPs that is involved in this splicing process, that is U1, that is U5, U4 and U6, they also attach themselves to the introns at this stage, resulting in the complex known as spliceosome. So here in the diagram you can see, it will make you more clear. So here this U1, it is being attached to the 5' site, whereas this U2, it attaches to this branch site which is near the 3' end. It is not the 3' end because 3' end is basically the acceptor site. So it branch site is near it, I have told you before. So this U2, it has affinity for this U1, and as a result it will attract this 5' site. Then what happened during this process, the other uRNAs, these one U5, U4 and U6, they also attach themselves at this stage to the intron, and as a result a structure known as spliceosome is being formed. So this is the spliceosome. Okay, and after that the cutting and joining reactions takes place that excises the introns and they ligate the exons, and these reactions of these cutting and excising, they takes place within the spliceosome. And once the process of this intron splicing, it is being completed, this spliceosome, it dissociates itself, its components, that one snRNPs, and they splice messenger RNA is released. So what will happen? Again, if I repeat this process for you, U2 will join at the 5' region, U1 will join at the 5' region, U2 at the branch side. Then what will happen, this U2, it will attract this 5' region and it will form a ligate structure, and the other RNA that were uRNAs that were U5, U4 and U6, they will also attach to the intron, and this base structure is basically known as spliceosome. And here the cutting and the ligating will take place, and after this all process which takes place inside the spliceosome, these all uRNAs, they will be released, these components snRNAs, they will be released, and the two ends will be joined, means the two exons will be joined, and the intron splicing. This was the entire process of intron splicing. Here it is in color form. You can see the two exons, and in between there is an intron. And what happened is that this intron, it will form a ligate structure, and this ligate, this structure, it will be removed, lariate structure, it will be removed, and these two exons, they will join together. So this is the process. So this was all about the processing of messenger RNA. But what happens to the transfer RNAs and the ribosomal RNAs that are being present within the cells? So you know that transfer RNA and the ribosomal RNA, they are structure molecules and they have roles in protein synthesis. However, these RNAs, they are not themselves translated. Okay, in eukaryotes, pre-RNA ribosomal RNAs, they are transcribed, processed and assembled in ribosomes in the nucleolus. They are known as ribosomes factory. So they are assembled there, while the pre-transfer RNAs, they are transcribed and processed in the nucleus, and then they are released in the cytoplasm, where they are linked to the free amino acids for protein synthesis. So we will discuss their processing one by one in detail. First we will discuss ribosomal RNA processing. So there are 4 ribosomal RNAs in eukaryotes, and these the 4 ribosomal RNAs, they are first transcribed as 2 long precursor molecules. Means 2 long precursor molecules are formed when 4 ribosomal RNAs are transcribed, and one of these precursor molecules it is processed into 5x ribosomal RNA, and the other one it spans the 28x, 5.8x and the 18x ribosomal RNAs. Then after that what happens is that the enzyme it cleaves the precursor into subunits corresponding to each ribosomal RNA. However, in bacteria there are 3 ribosomal RNAs, and they all are transcribed in one long precursor molecule that is later on cleaved in the individual ribosomal RNAs. Some of the bases of pre-ribosomal RNAs are methylated for adding stability as well, and mature ribosomal RNA it makes up to 50 to 60% of each ribosome, and some of the ribosomes RNA molecules are purely structural, whereas other have catalytic or binding activities. So these were the characteristics of ribosomal RNA. And however, in eukaryotic ribosome it is composed of 2 subunits. You know that the larger one is known as 60S, whereas the smaller one is known as 40X subunit, and the 60S subunit is composed of 28S RNA, 5.8X RNA and 5S RNA and 50 proteins, whereas the smaller one, that is 40S subunit, it is composed of 18S RNA and 33 proteins. Bacterial ribosome on the other hand, it is composed of 2 similar subunits, and with have these both subunits they are slightly different components. The bacterial large ribosome subunit it is 50X subunit, and it is composed of 23S ribosomal RNA, 5S ribosomal RNA and 31 proteins, while the small bacterial subunit is 30S subunit, and it is composed of 16S ribosomal RNA and 21 proteins. And the 2 subunits they join to constitute the functioning of ribosomes that is capable of creating proteins. So this was all about the ribosomal RNA processing. Now let's discuss the transfer RNA. So each different transfer RNA it binds to the specific amino acid and it transfers it to the ribosome. And the mature transfer RNA it has a 3-dimensional structure through the intermolecular base pairing to position the amino acid binding site at one end and the anticodon in an unbase pair loop of the nucleotides at the other end. And it has the anticodon is a 3 nucleotide sequence that is unique to each different transfer RNA that interacts with the messenger RNA codon through complementary base pairing. You can see the anticodon site here where it is written AAG. So it is basically unique for each transfer RNA. And here the base pairing, complementary base pairing occurs between the messenger RNA and the transfer RNA. So this is in the diagram you can see it is a 3-dimensional structure of the transfer RNA. Now let's discuss the steps that are involved in processing to convert the pre-transfer RNA to a mature transfer RNA. So 5 steps are involved to convert the pre-transfer RNA to the mature transfer RNA, and the first step is basically the cleavage of 5' sequence. The 5' end of the pre-transfer RNA is called the 5' sequence and it is cleaved off. The second step is the 3' end of the pre-transfer RNA, it is cleaved. The third is that the addition of the CCA sequence, and in all eukaryotes pre-transfer RNAs but in bacteria only few, in some bacteria and archaeal pre-transfer RNAs, a CCA sequence of nucleotides is being added to the 3' end of the pre-transfer RNA after the original 3' end of it is trimmed off. We have discussed in the second step is the trimming of 3' end. So after that the CCA sequence of nucleotides is being added to this site, and this CCA is basically the site at which the transfer RNA amino acid will be added later on. The third is the chemical modifications of the multiple nucleotides in the pre-transfer RNA. On average about 12 nucleotides they are modified per transfer RNA. This was the fourth step. The fifth step is that the trimming of the splicing of the introns. So the fifth step is splicing of the introns, because in significant number of eukaryotic and the archaeal pre-transfer RNA have introns that have to be spliced out. We have discussed before in the messenger RNA while we were discussing that the intron splicing is necessary. So here too, these introns have to be spliced out, and these introns, they are rare in bacterial pre-transfer RNA, but they do occur occasionally and they are spliced out. After processing the mature pre-transfer RNA, it is ready to have its cognate amino acid attached, and this amino acid of the transfer RNA, it is the one specified by its anticodon. We have discussed before that the amino acid, it attaches to that. Okay, so attaching this amino acid is called as charging the transfer RNA. In eukaryotes, the mature transfer RNA, it is first generated in the nucleus and then exported to the cytoplasm for charging. So this was the processing of the transfer RNA, and I hope that you have understand what I have told you in this lecture, and all concepts, I hope they are clear to you. So these are the references from which this data has been collected, and if you want to read further, you can consult these sites and the books that I have mentioned. Thank you for watching. Allah Hafiz.